So we're gonna present the professors ourselves.
Just say yes, an extra. Why not? We put them. Oh, you did. Okay. Yeah, just say hey. So next time. Okay.
Just so will you say your name, please? Okay.
Okay, and then just ask her to show.
Do you know how to kill rats? Just use that thing. Or you can do it manually. You can do it manually. Yeah.
It's fine. Yeah, you can.
Okay, folks, we're going to start now. First off, we got Dr. Jenny Silver. He's a senior lecturer in the School of Biological Sciences. He teaches a lot of courses that many of you are going to have, like parasitology, vector diseases, and a lot of stuff. And others. So I'm just going to let him take the floor.
Right, so thank you very much for the invitation. I've got a rough idea of what I'm doing here, but not completely clear idea of what I'm doing here. And I think that my understanding is that we've been off campus for so long. And one of the things I found is that I don't know, I'm trying to sort of see how many of you I recognise, but because we've been teaching off campus for the last 2 years, we've not really seen many of you. So I keep on meeting students in my 4th year class because I'm convener of the honours projects, saying, who are you? It's probably the first time I've met you. And it's the same with my personal tutor group. So I think part of the purpose of this is just so that we can sort of get to know each other again back on campus. And then the second reason that I thought I would take the opportunity here is to see what we can do to help you catch up, I suppose, with lost opportunities over the last couple of years, because there certainly have been quite a lot of lost opportunities. Having said that, I think that it's fair to say that in Aberdeen we've really worked very hard to keep as much on campus as we could do after the first part of lockdown. We did a lot more than many other universities in the winter of last year, and we've tried to keep open a lot more practical and project opportunity than I know has been available to many other students in other institutions in the UK. So I think we can sort of slightly pat ourselves on the back for that, but having said that, I think we can all admit that it has been a pretty stripped down experience. So I really want to talk about one thing that I'm very keen on, which is the opportunities that you have to develop your skills, practical and theoretical, through extra activities such as internships, placements and volunteering. And so I'd like to talk a little bit about that. Just before I do that, I'll just tell you a bit about myself. So I'm a parasite, I'm an immunologist who studies the infection biology of parasitic diseases, really, and some epidemiology. And my background is actually, I trained as a biochemist a long time ago, and I got into parasitic disease immunology by a series of fortuitous coincidences, and it kind of led me down that path. And it's been pretty good. I've had some fantastic opportunities over my career. Probably the best sort of decade for me, or the best decades were the sort of mid-90s through to the end of the 2000s, where I was involved in a lot of field work, particularly in sub-Saharan Africa, studying the immunology and epidemiology of a disease called sleeping sickness or African trypanosomiasis. Right now, my research interests are much more parochial, and my lab largely works on tick-borne diseases transmitted within the UK and Northern Europe. So I'm not really here to talk about research, though. What I wanted to talk about is these extra opportunities. And certainly in my career, for me, the springboard for getting into scientific research was an opportunity I had before I went to university to take my first degree. I was fortunate enough to get a placement, a work placement for 8 months in a company that was not far from where I grew up in Wilmslow, well, I grew up in South Manchester, in Wilmslow, Manchester, and called at the time ICI, nowadays it's called AstraZeneca. And so I had the opportunity to work in chemistry research laboratory developing new drugs, cutting edge drugs. In fact, I worked with the lady who developed the drug Nolvadex, which nowadays is called tamoxifen, and has been hugely important in the treatment of breast cancer and saved many millions of lives through that process. And so I had that great opportunity before I went to uni, and as a consequence of that, I managed to spend every vacation while I was a student working back in the lab there. And that really gave me massive experience, paid me wages well, and a level officer. And so I'm a really great advocate for trying to get these additional opportunities if you can. So we have an area in the Blackboard in my Aberdeen called the Biological Sciences Undergraduate Student Information Organisation. I don't know how familiar you are of it, but I've noticed in previous years not that many students know about it. So I'm just going to take you to it. I'm not going to specs on, am I screen sharing at the moment? I'm not. Okay. I should have brought my glasses. So we need to go to. Sorry about this. We use Teams all the time, but it is sharing fine. Okay, so hopefully you can see this. Okay, so how many of you are familiar with the organisations page in Blackboard Ultra? Hands up, let's just see. So that's about typical, about half of us. So we put a lot of stuff in here, so it's worthwhile being aware of that. You want to look for this organisation here called Biological Sciences Student Information for Undergraduates. And if we click on that, we've got various bits of content. It's all changed a bit since the move to Ultra. The area that I want to talk about is down here. Careers, volunteering, go abroad, internships, finding and funding, and the BI 487 mini internship course, which I'll talk about. And there's an area here on student societies. I've not looked at how well that's kept up to date. So let's just click into this area. Okay, I have no idea if this is being used or not, but there's an area for student societies and clubs. Do you know if that's being used? Shall we have a look? There does seem to be stuff for all the school societies in here, but that's not what I'm here to talk about. be aware that's there. And there's an area called Go Abroad, which is hidden from participants, I think because it's not been updated and it still dates back to the days of the Erasmus scheme, but I will see if I can find out what's going on with Go Abroad in there. Obviously that's relevant to those of you who are in first year, primarily. There's an area here on careers and volunteering, which actually I've not set up. I don't know who put this in here, and it's got a bit of information about some of the local volunteering organisations in conservation in particular, and some of the environmental charities that offer volunteering opportunities. And normally, around this time of year, we would run what we call a volunteering fair in the Zoology foyer. It's really been impossible to organise it this year because of all the restrictions on who we can invite in, and having large gatherings in the foyer, where we would bring in representatives from the various groups, where you might have the opportunity to do some volunteering in your weekends in or close to the city. And then there's this area here, which is the bit that I look after, which is called professional development in the workplace, placements and summer studentships, finding and funding. And so I'll click on that. I'm not going to go through all the different panels in here. You can read them at your leisure. But basically, there's some background information here, which is really basically encouraging you to read this stuff and think about how you might be able to use these opportunities to enhance your CV, to find opportunities for activity you might do in the summer vacation primarily, so between year 1 and year 2, between year 2 and year 3, and between year 3 and year 4. And then there's various directories which we keep, and we try to add to them, so they vary in real time, hopefully. One of them gives you lists of potential sources of funding, whereby you can get a stipend, for example, maybe research costs for doing a research project in an academic laboratory or in a research institute, a laboratory within Aberdeen or elsewhere. So have a look through that list. There's a second list, which is actually formal internship opportunities. Some of these are within biological sciences, some of them are more generic. We include in that list of internship opportunities things like Santander internships and others that I can't remember the names of at the moment, and I don't really want to open that page because I'll overrun the time, but please have a look through that. If you're not looking to get a job, there's various specialist summer schools that are listed here in the third panel. The summer schools vary. At the moment, most of them are in the area of molecular biology, but there's one British Ecological Society summer school, and there's a plant science summer school listed in there. And I'm hoping we'll be adding to these lists of summer schools over the coming month. And then there's a final panel here, which basically encourages you to think about joining relevant scientific societies, professional institutes, and the ones that are particular value, I think, to you, depending on which direction you're going in terms of your academic specialism, are the Royal Society for Biology and CIDE, the Chartered Institute of Ecology and Environmental Management. Both of those are really good because they are chartered institutes and they lead to qualifications which you can start building up experience for still as students, and at the same time they offer very highly discounted student memberships and grants for all sorts of things that you might support you, such as travel grants and subsidised training courses. So certainly think about joining a scientific society or professional institute, and the lists of academic staff associated with each one who can advise you more on those societies and institutes. And then there's a final panel here which I've got from Janice Montgomery. And so Janice is the careers advisor, the senior careers advisor for biological sciences. How many of you met Janice?
Online.
Yeah, well, online, including online, not necessarily face-to-face. So a few of you. So please take the opportunity to take advantage of what's offered by the Career Service. And so there's some further advice on internships, particularly things like management internships within the university, which again might be of interest in that panel there. So that's all a bit of a pep talk about trying to get opportunities for internships and placements. We want to look for a research kind of internship in the university in biological sciences or in one of the other schools such as medical sciences. It's never too soon to start looking. And in fact, when you look at some of these scholarship schemes, some of these summer school application deadlines, the deadlines are coming up quite soon, January, February. So you need to start working now and read up, ask, and I've got I implore you in one of these panels, just keep asking, keep hassling myself and colleagues. We won't get upset if you're keen and you're trying to get information on opportunities. We might say we don't have any, but we'll say maybe you can talk to so-and-so. And so please don't be backwards coming forward on that matter. It's part of our job to make sure that you have the best outcomes out of your degree. And so we love it when we get keen students looking for opportunities to do additional work and research in our laboratories. So that's part of what I wanted to talk about. I'm not going to go through all of those, but please read about those and tell your friends about them, because that resource is there to be used. Now, for those of you who are in third year, who's in third year? Okay, right. So there's a course that is available to you that you take in fourth year formally called BI 4027, and it's called Professional Development in the Workplace. And it's really a course you build yourself, and you build it around a placement or internship that you get in the summer coming up. So basically what it is, it's an opportunity for you to reflect on the graduate attributes that you develop as part of whatever activity you do during the summer. Now, we've had people doing BI 4027 that have done everything from working in the lab in the University of Aberdeen to doing environmental team for big cosmetics companies to working for whatever the guys who were called, I can't remember what they're called now, the Nuclear Decommissioning Authority, but they're doing great. So yeah, I'm getting mixed up. So have a look at BI 4027. There's going to be a presentation event on BI 4027 a week on Wednesday at midday, and I'll be circulating all third years information about that tomorrow. It will be recorded though, so if you can't make it, then that's not a problem. Again, you want to start thinking about planning for your vacation activity next summer. It will come around really quickly. So that's really about it for me. Any questions? Stun you all into silence. Yes, sorry.
So when we are looking for kind of a summer research that we could do in the SBS or Zoology department, and we go to the professors, like what we should like just ask them if they have any ongoing research that in which we could help him, or we should prepare somehow like what's going on and ask directly, like if we can help in a specific project, or like how is it the easiest for professors to give us opportunities, or.
So are you asking, should you come with a plan? Here's my project plan, can I do this with you?
No, but what's the best way to approach a professor in this kind of situation?
Just do it informally. Just say, I'd really love to work in your lab or research group this summer. What opportunities are there? What can I do? Now, it may be not everybody's going to have capacity this summer. So they might say, well, I'm sorry, I really can't do anything for you. But hopefully my colleagues, certainly what I would do is, if that's the case, I'll say, what are you interested in? What are you trying to get out of this? Do you need it to be in Aberdeen? Might you want to work elsewhere? And then I might say, well, have you thought, maybe you should contact X or Y. And so I might be able to put you on to other people you might on. And hopefully my colleagues and rest of you, would you support that? We'll do our best. So in some cases, when we get an approach, we'll say, yeah, and this might be a really good project where we might be able to apply for a Wellcome Trust summer studentship or something like that. And that'll be even better because I'll get a sort of research cost into my lab, and you'll get a stipend for doing that work. So that's where it's worthwhile having looked through that list of scholarships and studentships. Because equally well, you might say, well, can I apply for a Gurdon studentship from the British Society for Development of Biology, for example, be able to work in, you might go to Zoology State, so I can work in the lab. I would like to apply for a Gurdon fellowship. So have a look through that list. So it's up to you to do the legwork. So you need to show you need some initiative here. We can't make it happen for you, but you can make it happen for yourselves, and that will be really good for your careers. Okay.
So next up we have Barbara Cini. She graduated from the University of Aberdeen in marine biology, and now she runs the project in the field lighthouse field station of Aberdeen. So, Barbara, can you hear us?
Yeah, sorry, it was a little quiet there.
Can you try sharing your slides if you have any?
Sorry, I still can't hear very well.
Oh, okay. Sorry, I thought you could.
Okay.
Can you hear me now?
That's a little bit better.
Okay, sorry.
Do you want me to get started?
Yeah, if you can.
Okie dokes. Can I share my screen? Is that possible? How's that for everybody?
Yeah, that works.
Yeah, great. And can everybody hear me okay?
Yeah, I can hear you.
Cool. So this might be a bit challenging with the sound, but what I thought I would do is give you guys a little bit, because you probably don't know me or potentially don't know me, so I thought I'd give you a little bit of the background as to who I am and what I do, and then really open it up for you guys just to ask some questions for anything that you're interested in, whether it's you're interested in the Lighthouse Field Station where I work, or whether you're interested in how I got to where I am, or yeah, anything about the work that we do. So if you guys haven't heard about it, the field station is one of the field stations, obviously, the University of Aberdeen, and we're based up in Cromarty, and we really study marine top predators. My job specifically is to look after the long-term bottlenose dolphin project, which is an individual-based study, and we mainly use photo identification to recognise the individual dolphins. So you guys probably know this already, but for anybody who doesn't, we take photos of the dorsal fins of the dolphins, and we recognise the individuals from these natural marks on their fins. So they have, when they play and fight with each other, they make these marks using their teeth. So you can see on this guy here, there's tooth-rake marks, and there's also nicks, which are bits missing from the fin. And what we can do with these is we can actually basically follow individuals through time. So there's been dolphins that we've known right from the start of the study, which was over 30 years ago, and we can keep track of them. We can also keep track of individuals related to them, so their calves. And then we've actually got 3 or 4 generations now because we've been doing this so long. And what that allows us to do is it allows us to look at population dynamics, so we can estimate abundance, survival, reproduction, and it really allows us to monitor the population to see how well it's doing or how badly it's doing. And the population here is in a special area of conservation, so it's obviously very important to be able to monitor this population. So that gives you just a very brief overview of what I do. In terms of myself, I actually studied as an undergrad at Aberdeen, like yourselves. This is actually a second career for me. So to start with, I actually used to work at British Airways and realised that was not what I wanted to do with my life, and came back and studied marine biology in Aberdeen as a mature student. And then I did my honours project here at the Lighthouse with Paul Thompson, who runs the place, and then basically just kept coming back until he gave me a job. And so I've been here now for 16 years. I've been here, can't quite believe it, and just working on different projects. And then I did get this job running the bottlenose dolphin research. And then also I've done my PhD, so I did a part-time PhD here at the Lighthouse on the project, and completed that back in 2017. And yeah, I clearly love what I do, so that's why I'm still here. So did anybody have any questions, as I say, whether it's about the project or myself or advice or anything?
I do have a question. Yeah. So when you were doing your dissertation with Paul Thompson, did you do like an internship with him during the summer, or was it just the fieldwork part of your dissertation?
Sorry, Patricia, I didn't 100% catch that.
Oh, okay.
So if you wanted to do an internship here at the Lighthouse, is that what?
Yeah. So if when you were doing your project, like your honors project with Paul Thompson, did you work there as part of an internship with SBS, or was it just like the field work of the dissertation itself?
Yeah, so actually I was doing my undergrad in marine biology at the university, and obviously part of that is to do the honours project, as you guys know. But I knew I wanted to. I didn't know exactly what I wanted to do, but I knew I wanted to work with marine mammals, and obviously Paul was the dude to work with. So I actually got in touch the year before I was to do my honours project and asked if he needed any help. And so I came up and I volunteered for about a month here. And while I was here, I talked to everybody that worked here. There was, a few people doing PhD. There was a few master's students at the time actually as well. And I really just kind of picked their brains as to different things. Like if you were doing an honours project, what would you do? And what do you think we still need to know about marine mammals? And really tried to get some good ideas. And then I actually came up with my own honours project that I talked to Paul about, and he said, yeah, that's a good idea. It really helped by people I'd talked to. And then, yeah, so he let me do the honours project. So I didn't enter or work here or anything, but I think just being here for a month, like meeting people and finding out what they actually did and what was still maybe unknown about the dolphins really helped for me to come up with this honours project and be able to carry that out.
Okay, that's very cool. I knew about that you could volunteer in the Lighthouse, but they weren't taking anyone last year because of COVID.
Yes.
Oh, sorry.
So at the moment, I would say that's the same. Unfortunately, I'm not sure how likely that is to change for next summer. So what we normally do for the photo ID field work is we have our own boat here, and we go out with probably we tend to try and go out with 3 people on the boat. One person driving the boat, one person taking photos, and the other person helping with notes. And then having 3 people allows you to have a really good coverage, so that you have good spotting of the dolphins when you're out. But for the last few years, we've only got 2 people on the boat, because that person tends to be, a student that's up here, or volunteers that come and help, or interns that come and help. But we've just not been able to do that, as you say, because of COVID. And at the moment, the plan is probably to stick to that next summer, but we're hoping it could change. So anybody that's kind of interested, or feel free to keep in touch with us. And we could definitely let you know if that does change, and hopefully it will. Hopefully things will get better, and we'll be able to have people come and help us out.
Okay, that's very cool. Thank you. Does anyone else have any other questions? I'll repeat them over here. No?
There was just before I run off. Can everybody still see my screen? There's so I don't know if everybody knows, but we've been doing a kind of promotion, I don't know what to call it, a social media campaign for the last nearly year now, actually. There was an International Women in Science Day last, I want to say February, seems a while ago, and we've been, you can imagine there's been, over the 30 years, there's been some amazing scientists, both male and female, that have gone through the Lighthouse doors, and they've gone on to do, have some amazing careers and really inspirational people that have inspired me. And what we did is, specifically looking at the women, because of International Women in Science Day, we actually asked some of these women to write about themselves and to tell, how did they get to where they are, and to give advice to kind of budding scientists. And this is on our website, so I'd really, for the guys as well, not just for the women, I'd really advise you to I think it'd be really helpful to have a look at that and just get some viewpoints from, there's people that have worked all over the world now and are really doing brilliant careers, and even in different careers, not necessarily have stayed in science, but maybe give you some other things and some other advice that might be really helpful.
Yeah, that's very interesting. I've seen the post in social media lately from the
Great. Cool.
Yeah.
And everybody has a slightly different viewpoint, so I think it's interesting to meet some of those profiles.
If no one else has any questions, then yeah, I think we can wrap this up. And
that's okay. I hope that was helpful, and thanks for including me.
Yeah, thank you for coming.
Okay. Bye.
Next up is Stuart. Yeah. Yeah, so he works with genetics and evolution. You probably you're gonna do a course with him in third year. But yeah, he has a lot of cool research in deep sea as well. Yeah. Yeah. Sure.
Just shaking the chat like everyone can hear it.
I asked before, but no one answered, so that's why I thought it was working.
Right. All right, cool.
So,
Evening, everyone. So my sort of 2-minute spiel sort of follows nicely on from Jerry's earlier on. What I'm going to do is just talk a little bit about some of the opportunities that have been available in my lab over the past few years, and sort of flag it in terms of the sorts of research that I do. So I recognise some hard faces, don't recognise others, so maybe you don't know who I am. I'm Stuart Pinn, so I'm the evolutionary biologist and molecular ecologist within the school, and also coordinator for animal evolution biodiversity. I was originally a marine biologist, and I got into evolutionary biology almost by chance and by some very kind favours by a couple of eminent professors in the places that I serve. So I'm very keen. What we try and do as members of staff is try and encourage you lot as much as possible to have those sorts of opportunities. And we're especially aware over the past 12 months or so that it's very, very difficult with all the limitations around COVID. So the more we can do to get you back on track, the better, as far as I see. So I think we just want to explore some of those opportunities and just flag up a few ideas. So as I say, my work is on evolutionary biology, and I've been lucky enough, because I was at the sort of at that point when they started realising you could use molecular tools, that they started throwing those molecular tools at all sorts of things. So I could work on birds, mammals, invertebrates, all sorts of things. I went across almost the entire spectrum of taxonomy, if you like. But more recently, I've gone back to the bits that I like, which is primarily deep sea research and more marine biology-based research. I'm a big fan of that, that's really good. And so a lot of the work that's coming through my lab at the moment is sort of focused around that. And I put this slide together just to highlight some of the things that actual students have used to get slightly more experience around all of that. Now, with molecular work, it's expensive stuff. And so what we need to, we can't sort of just invite loads and loads of people into the lab. But what we do generally do is try and sort of funnel it through the honours project and an extended honours project. And I think there's a lot of opportunities for that. And there are now quite a lot of molecular biologists and molecular ecologists within the school of different flavours. And so I think if this is your sort of idea, this is your virtual boat, then I think there are opportunities within the school. So I've sort of organised this slide into a couple of different ways that you can't really make much out of. But at the top, there's a couple of the perhaps learning societies that marine biologists perhaps would want to focus around. So when I started out, it was all the Marine Biological Association. And something like the Marine Biological Association has their own journal, and so you can stay at sort of forefront of research in that area. And more frequently, more recently, because I've become a bit more genetically focused, there's things like the Genetic Society and the European Society for Evolutionary Biology. And I would really suggest that you sort of look at the web pages of some of these learning societies, because I think as well as perhaps advertising some opportunities for internships and funding support, they're also a great place to learn who the main players are within the field, and also perhaps some of the latest sciences coming through. So depending on whatever your particular interests are, there will be a learning society that is appropriate. So as I say, mine, what I keep an eye on, things like the Marine Biological Association, Genetic Society, European Society for Evolutionary Biology. The Deep Sea Biology Society is quite young, but it's very, very dynamic, and a lot of work gets sort of flagged and opportunities get flagged up there. So that's another one to think about from that perspective. And then Schmidt Ocean Institute is a group that are increasingly into outreach and student experience. And so to a large extent, keeping an eye on groups like Schmidt Ocean Institute is actually quite a useful thing to do. And the reason I put all of those up there until we get down to the Royal Society of Biology is that all of those are funded an honours project student within the lab. And so it's well worth you keeping an eye out for all sorts of opportunities there are in keeping around. Jerry flagged up that the Royal Society of Biology is a great accreditation service. What they've also got on their website, which is what I've just flagged up there, there's a whole list of different graduate studentships that are available, primarily from second to third year if you're down in England, or third year to fourth year if you're up in Scotland. But again, there's an awful lot of very, very good opportunities there, and I really recommend you have a look at those, see what's going on. Carnegie Trust do very similar things to Scottish students and Scottish universities. And one that you perhaps don't know is quite new is that we do a lot of our PhD training through what known as doctoral training partnerships. And the school is associated with one called Quadrant, which is an alliance with Belfast University, Bioscience and Geoscience. And there's also one called Super, which is all around some marine biology within Scotland. And associated with both of those is a thing called REPS. And REPS is this research experience placement scheme. And that is to allow undergraduates to buddy up with a PhD student for the summer. And just like all these other schemes, there's some great opportunities there. And it's to a certain extent funding. But what you might want to be thinking about is how you can dovetail what you might do in that summer with what you could do the subsequent semester in terms of your undergraduate project. Because if there are opportunities for getting slightly more experience in all of that. I also just want to flag up a couple of bits and bobs on that right-hand side there. And so I talked to my animal evolution biodiversity class about this, about whether you've got a professional social media presence. And this comes in two reasons for doing that. One is that it allows you to get your name out there in terms of having a network of contacts who may well be looking for a master's student, PhD student, internship student, all these sorts of things. But also, it's also a great way of you staying on top of what the very, very latest research is. If you can work out, if you follow the right people
and you follow people right at the forefront of the field, then you know what science they're coming out. And as a consequence, you're that much better informed for anything that you do moving forward. And so I would certainly suggest that you set up a professional Twitter account, and then you just follow within that the people who are associated with the sorts of science that you're interested in. So I think you get an awful lot out of that. And then as you start thinking about developing your CV and what's happening on graduation, having a LinkedIn site is another great way forward. You want to get your name out there, and you want to have that network of contacts so that you know what opportunities are out there. And then I think the last thing I want to say at this stage, and this is me sort of sat on the other side of the table in terms of doing PhD interviews and master's interviews, is that don't think of gaining this experience as sort of as an exercise as part of passing the course. You should be doing what you want to do and what enthuses you. Because you might, if you go for a molecular project, just because you haven't necessarily had any molecular experience doesn't mean that you're then below the pecking order of someone who has. You know, what we really look for when we're doing these sorts of interviews with PhDs and masters and when I start on job interviews is people who are articulate and enthusiastic and passionate and can actually make the point that they want to make and highlight that they understand the system that they're working with. So it's not just a case of ticking a few boxes of, like, yes, I've done a PCR, yes, I've done this, yes, I've done that. It should be a case of you get experience around the things that you really want to do. And in some cases, that can be some quite bizarre stuff. So I stuck the R logo there. I don't know whether any of you know R or whatever, but there's an awful lot of opportunities for self-learning. So if there aren't any opportunities to go and live in a lab for a summer, if you spent it, just spending an hour a day thinking about how to use R. So if someone says, oh, confident moving on, you say, yes, absolutely, I've done this, I've done that, I've done this. So you can gain an awful lot by a little bit of self-teaching and self-directed research in terms of you understanding what's going on. Yeah. And that's really how I started, because a comment was made by the big sort of marine biology identification guide by a guy called Ryland and Hayward. And John Ryland said to me, no one knows anything or very much about how to identify amphipods. So I spent my summer trying to work out how the bloody hell to identify amphipods. And I got a PhD as a consequence of that. Not because of any other reason, apart from I could just wax lyrical about amphipods, and that was just obviously what the focus of that particular PhD was. You know, and that comes again. So one of the Smithsonian Institute, they're a big philanthropic group, and there are about there are 3 people in the world who know anything about deep sea amphipods. Yeah. One is a lady down in NOC in Southampton. One is a lady down in Niwa in New Zealand. And the third is a guy called Ryan Eustace. And Ryan was an honours project student of mine about 2-3 years ago. Yeah. And Ryan decided, there's an opportunity, I don't know what no one knows what we're talking about, I will learn it. He became headhunted because he chatted to these people in his social media network about how to identify these amphipods. And Ryan then he's now a well-known international figure just because he decided, actually, I find this quite interesting, it's something I've got an opportunity here. And we can help you along with all of that. You know, if there's anything that you want to do in some of that self-learning opportunities, even if we can't offer you a placement in the lab, we're more than happy to help in terms of saying, yes, perhaps go down this route, perhaps go down that route, perhaps try this, perhaps try that, perhaps talk to this person who might not be in Aberdeen, but would certainly be willing to help. You know, I'll finish by echoing what Jerry said, is that please don't ever think that hassling us. You know, this is part of our job, it's something we enjoy. And we get an awful lot out of seeing you lot progress and become more competitive upon graduation and going into those positions of influence that ultimately is what we want to do. So we're more than happy to help you out. Please don't hesitate to share. Happy to take any questions. Yeah.
You spoke about R.
Yeah.
What is R?
So I just took R up. So R is a programming language. It's the new way of doing statistics. So, you know, you may well have been taught about Minitab or MATLAB or whatever, but the way some research science is done is using a line-driven, command-driven programming system. And R is sort of everyone can talk. Maybe the Python, that's the one from a bioinformatics perspective at the moment. And if you think about it, if you spend half an hour a day, every day for a week, for a month, for a year, you'll become an expert at something. You know, it's just about having that little bit of drive and that little bit of attention to detail. But, you know, don't underestimate what you can deliver in terms of a bit of self-learning.
Thank you.
I know that internships are usually taken in second, or like between second, third, and third and fourth year. But given the dynamic over the past 2 years, are there any opportunities for fourth years who didn't have any internships to still get some kind of experience like that?
Yes, there are. It's difficult. The bottom line is that, as Barbara sort of highlighted, we're not back to normal yet. And so limitations in the lab means that it's more difficult. But there are still some opportunities, and even beyond from graduation, there's still those opportunities there. So just because you sort of finish doesn't mean that it's the last one to see you. It's a case of you can apply and there are opportunities to do work beyond that and go for it. As Jerry highlighted, an awful lot of resources where people are supporting studentships at various stages of the career. So keep looking at those and go from there.
Also, I realize it's not a quick question, but when he decided to get good at identifying these deep sea animals, how did he go about doing that if there's not any information on it?
Exactly. So what he did was he learned about how you would identify shallow water species, and then sort of that knowledge to think about, well, that's slightly different down there, that's slightly more, that's slightly less. But it was just, everyone was amazed by how much you could actually achieve and what he did achieve. The fact that it opened so many doors. And the point is, don't underestimate you lot as individuals and authorities. Just because, I keep saying this, with just an undergraduate, that's an absolute nonsense. You can be as big an authority as an undergraduate student as you are as a postgraduate, as you are as an established professor who's been at it for years. It's just that your level of experience is not quite at the same level as this, but you just gain experience by doing it. Cool, right?
Thank you very much.
So if anyone wants to go and see more, we're gonna come back here at 8 o'clock. So you have also we have Open Society stalls next door. So if you want to ask us if there's any opportunity.
Next up, welcome Dr. David Fisher, who's going to explain social interactions and the role they play in animal evolution and ecological process.
Thank you. Can you folks hear me at the back? So, are you having a good time? Are you having a good time? Well, that ends now. My name is David, and this talk is about R, coding, and statistics. Now, if you lot are anything like I am and my cohort when I started my undergrad degree, I took biology and zoology specifically to get away from having to do mathematics and things like that. And so imagine my horror and the horror of almost everyone else in my cohort when there they are again, those horrible numbers come back to haunt you. They really are often like this scary monster and everyone dreads those statistics lectures. Not everyone, admittedly, but a lot of people like cannot be bothered to drag themselves out of bed to go to that statistics lecture. And especially, this is especially bad when it's combined with learning how to code, which doesn't always happen, but sometimes you're required to learn this new programming language, something like that, to be able to then carry out your statistics. You're being taught two bamboozling and disgraceful and disgusting languages at once. You're like, what is going on here? This is awful. This is horrible. But actually, I think this is a really, really great opportunity that you can kind of grasp the nettle and learn these skills, slay the beast, and then you can learn incredibly useful transferable skills, not just to allow you to kind of do lots of different jobs or opportunities following your degree, but also within biology to ask and answer all sorts of different and interesting questions. Now, I know some of you don't get the opportunity to study something like R during your degree, but there are an enormous number of free resources out there if you are interested in giving it a go. So I particularly like Swirl because it teaches you R in R. So basically you download Swirl, fire up the package, and then it'll essentially just ask you to do things entirely within the R environment. Now, this is just one particular example. There are lots of different things out there because R is free and open source, and so everyone is using it all the time in all different countries, and everyone's helping each other out and creating lots of different resources. But I like this one, and I've had about 3/4 of my honours project students so far have started without having used R before, and they've all used R for their final projects, and partly because I got them introduced with something like Swirl, and partly because I can just teach them the basics, because it really isn't that difficult when you have someone kind of showing you the ropes and helping you out a bit. And if once you get rolling and you get a bit of a feel for it and you want to like try out new things and so on, then there are an enormous number of opportunities out there. So often when people publish papers, so this is one of my papers, as the introduction said, I study social interactions mostly in insects. This is a really cool paper I worked on New Zealand giraffe weevils. Now, they're not quite like giraffes. They don't have a really, really long neck. They have a really, really long head, to make it look like they have an enormous neck. They're absolutely ridiculous looking things. And actually, we made a front cover. I don't know if you can see this, but yeah, it's super cool. I've never been to New Zealand, unfortunately. My little sister is actually stuck there and has been there for about 2 years. Can't get back because of the pandemic. So I'd like to go to there to see the weevils and also see how she's doing. But I saw Christina Painting, the senior author, give a talk at a conference. And I went up to her and I said, that was awesome. This is the coolest, one of the coolest study systems I've ever seen. I want to get involved somehow. We ended up chatting, and because I had good quantitative skills, I was able to suggest a new way to use her existing data set. So I got her existing data set, collected in 2013 for a master's project, and reanalyzed it, and then got this paper out of it. And what people are doing even more now as well, and this really helps you folks out for then improving your own skills, is they are publishing, they are putting the R code and the data to recreate the results of the paper in the supplementary materials. So this paper, I'm a bit of a nerd about this, why I always do this, so mine's a good example, but more and more papers are doing this. You can find the exact R code to recreate this analysis along with the spreadsheets with the original data in to then redo the analysis yourselves. So that is useful for a better understanding how on earth they did what they did in the paper. but also by just, you can copy and paste bits of this and use it for your own work. So you can suddenly you've got all these skills because you're just using them from here. And then you can go through this and you can pick up suggestions and ideas and just practice using your R skills. If it's, and you're going to be motivated to do this because it's exploring something you're interested in, which is this cool paper you like, and so you wanted to go and find out how they did it all. And so this doesn't work for older papers, certainly, but the stuff published in 2020, 21, 22, more and more of them will have the R code and the data freely available for you to just give it a go. And as I said, so this stuff is often free and it's out there. It just takes your time and your passion to go out there and make it happen. You need a decent internet connection most of the time and a computer, but nothing particularly fancy. And these skills you can pick up are almost always incredibly valuable. So you can learn these quantitative skills, these database manipulation skills, and they can open up all kinds of doors for you. So I knew a PhD student called Cameron Anderson. He was at the University of California. And he was studying huntsman spiders for his PhD. So these are they're sometimes called daddy longlegs, they're sometimes called other things. They are they're not true spiders, they have a single body part and their legs coming off it are really, really long. And they engage some interesting social behavior, some kind of primitive forms of maternal care in some groups. But he got pretty good at R and manipulating data during his PhD. And in the end, he decided he didn't want to stay in academia, he wanted to use these skills elsewhere. So do you want to guess who he works for now? Random guesses. Anyone? What was that? That's disappointing. Yes, for the first time. Well done. Yeah, Cameron now works for Google because he picked up these skills, analyzed data sets, and kind of, do statistical analysis to explore patterns and so on. That is incredibly valuable in all kinds of fields. I know also people, when I was doing a postdoc in Canada, ended up working for banks. I think that because they can manipulate data and they can ask and answer questions for people using these skills they've picked up. All these resources are free. So that's all I have to say, but thank you very much for listening. I don't know, organisers, how much time we have, if I can take questions, or if we, a couple of minutes, couple of questions. You can ask about the draft weevils, that's fine. Yes.
When you're if you copy and paste code from other R to use in your research, what would you have to do to make sure it's not plagiarism?
Yeah, great question. Yeah, great question. So it's an interesting one. The ethos of people using R is very much, we share the code, we basically give you permission to use it. And so if someone went into that paper and copied and pasted it and used it to do like social selection analysis in their own study system, I'd probably expect them to cite the paper somewhere because it's a relevant topic, but otherwise I wouldn't be worried about it. And I send people code all the time and I don't expect them to cite my work. Maybe in the acknowledgement that I've really helped out, but it's not really like copying and pasting text from Wikipedia, which is considered plagiarism, because you're kind of copying someone's tool use. It's generally considered it's fine. I mean, typically you can just cite them if you're copying a lot and you're really just directly building on what they've done. But if you're using it in a completely different context, I don't think anyone would worry about it. Yeah, but yeah, good question. Yeah.
So is R just a software or is it a language as well?
Technically, I think you refer to the R programming language. Yeah, it's called R because it's something similar to S. It's a really lame programming joke, I think. Yeah, so it only really, you can use R to create documents and PowerPoint and so on, but it's mostly used for doing data manipulation and statistics in the R software, yeah. But the way it works is similar to some other coding languages, so it's not like learning this is it only useful for R and nothing else, but kind of a general kind of way you can use stuff and commands and functions and loops and so on is applicable for kind of other things. Yeah. Anyone else? No? Wonderful. Well, thanks for your time.
connect this thing. Who knows how to connect this up? I need help with that. Okay, otherwise we transfer it maybe onto a stick.
Yeah.
Let's do that. Let's put that on the stick.
Yeah, that's fine.
Okay. Okay.
Don't worry, you're fine.
I'm fine with that.
Yeah, you're fine.
Yeah,
Don't
okay,
worry.
this is good. This
Yeah,
is
yeah,
good.
yeah.
Okay, use seems to work. Yeah. Okay. Just goes in here. Yeah. Okay.
If we can. Oh no, that's mine.
USB drive. This one here?
This one? No, should it be this one?
No.
USB
Try
drive.
this.
Oh, sorry.
This is it. This one.
This one.
On top. On top. Advanced presentation. Exactly. Okay.
Yeah.
Good.
And then if we can. Oh, I have to.
You can
open
remove
first.
my stick already.
Yeah. Not that one, this one. There we go. And then. Dun, dun, dun. Where is it?
Oh, yeah. Here we are. Okay, great.
Wait, I'm trying to share the screen, but.
Yeah. Share the screen with the online session.
Yeah, exactly.
Yes. This is what.
But I'm. Oh.
Test. Okay, good evening. Can you hear me? I believe so, yes. And you can see my screen, even better. Yeah, I'm afraid. What do you want to say a few words? Go ahead. Yeah. No, go ahead. This is even better if you do it. It's even better.
So our last speaker is Frigyes Cooper. He does a lot of research with Algin University, and he also teaches in some of the marine biology courses in second and third year.
Thank you very much. Yeah, okay. Frigyes Cooper, Chair in Marine Biodiversity, and today I'm going to talk about what is a tool in my group. I should say that most of our research, of course, is lab-based here at the University of Aberdeen. But often for getting biological materials, but also survey data, we rely upon scientific diving. So I'll say a few words about this tonight. This is actually a photo from an expedition to the Canadian Arctic back in summer 2009. What is happening here? I can't see anything now. Yeah, there's students commenting. What's happening here? I can't.
No, it's just the live captions.
Okay.
Yeah, so it's sharing. Yeah, it's fine.
So I move. Okay, so I will talk about the significance and potential of scientific diving. And I should especially acknowledge here my friends Hugh Brown and Martin Sayer, who work at what was formerly the UK National Facility for Scientific Diving. It was a NERC-funded facility. Unfortunately, it was decommissioned or whatever you want to call it a couple of years ago, but it's now a very successful private company that provides scientific diving services for anybody. Well, let's go a bit back in history. For scientific purposes, diving started in the 1940s, and it was in particular the work of Jean-Yves Cousteau, but also Hans Haas, yeah, they started working in the Mediterranean and the Red Sea. However, marine biology has started much earlier, arguably probably in ancient Greece and Rome, and there are names like Theophrastus, Pliny, and others. Theophrastus is also considered the father of botany. He is actually from the island where we had several editions of our field course, Mediterranean Marine Biology, the island of Lesbos in Greece. And yeah, then there was a new dynamic in the 19th century, and names like Darwin, Hooker, yeah, Dawn and Haeckel come to my mind. And Dawn is the founder of the Naples Marine Biology Station, which is the oldest marine lab in the world. But only in the 20th century, diving became available as a technique which put the scientist right into the middle of the community or the environment that he was studying. And yeah, a key argument that you will see like a red line through my entire talk is that being able to see, touch, and selectively collect in situ is a priceless advantage. And this is one of the reasons that I keep mentioning when talking to people who say, oh, we can do everything with submarines or ROVs, we do not need to send anybody down there. It's not the same thing. And yeah, this applies in particular to the seabed, to the benthic environment. And before diving, all the collections of specimens and data and samples relied on using fairly indiscriminate methods like bottom grabs or dredges, where you couldn't see. You had only after deploying such a device, you could see what you might have caught or not. But inevitably, a lot of things were overlooked and never collected. So that means that in many parts of the world where there has been little scientific diving activity, much of the marine biodiversity is understudied, and that applies in particular to some of the areas where we have been working, like the Falkland Islands or the Antarctic Peninsula, or more recently the Arabian or Persian Gulf. And yeah, even in the Mediterranean, actually, we right now have a PhD project in my group ongoing, that of Vivian Luisido, where very little is known about the seaweed flora beneath 30 meters depth. And yeah, even more, this applies to remote places like the Falkland Islands or South Georgia, and yeah, even pioneers like Carl Scottsberg, who is the eminent name for that part of the world, has overlooked a couple of things, because in the early 1900s, no diving was available, and everything he collected was brought up by dredge. The same applies for Ascension Island, which is another very remote place in the tropical South Atlantic, where we have been now 6 times. And yeah, our longest diving-based expedition, which was there in 2012, yeah, already 9 years ago, with 2 diving phycologists. Phycologists are seaweed studying scientists. Yielded at least 15 new seabird records. And yeah, that was, in other words, more than 1 record statistically per day of diving. And the same applied for the findings of our colleagues who were zoologists, yeah, studying fishes and marine invertebrates. And if you go to places like this and you start diving and you know something about the biodiversity that occurs in principle there, you're likely to pick up lots of things that are either new records or taxa that are completely new to science. And another argument is that the qualities of samples that are collected by diving, because they can be collected very carefully, is often unsurpassed by what mechanized tools like a grab or a dredge can achieve. And in yeah, many cases, such samples or data are of a quality level that is sufficient to be directly used for publications, or they can be further processed by other established techniques. And that can be anything in the lab that you can think of, like microbiology, culturing, molecular and cell biology, or biochemistry, or natural products chemistry. And also it has to be said in support of diving that it's relatively low or non-intrusive. It's low impact because it is not very destructive. You have a diver going in there and he or she may observe what lives down there and then very selectively pick certain organisms or samples. And yeah, there are also fewer conservation issues compared to pulling a dredge through a seabed system, which a dredge can be something like a bulldozer basically. And imagine this yeah, in a system of slow-growing corals or sponges or something like that. Yeah, also, diving is, deployability of divers is highly mobile and versatile, and it can often be arranged with short notice or little logistics effort compared to using a large research vessel. And in parts, you can even use tourist infrastructure that is available in certain parts of the world. And also, yeah, the ease of documentation should be mentioned. Nowadays, underwater photography has become very low cost compared even to the 1990s when I started this as an undergrad student. digital photography gives you a result in real time, and that is, in other words, parallel to the collection or an experiment. More significantly, a diver can go beyond just collecting samples. He or she can become the operator of an experiment, and that means setting up, fine-tuning, following, and recovering an experimental setup. And the capabilities of human hands, eyes, and in particular the brain, are hard to match by the technology that is available on automated devices like ROVs and benthic landers. And these are really the key tools that a diver has available. And up to now, there is no ROV with an onboard computer that can match what the human brain can of a scientific diver. And de facto, diving puts a scientist's brain operationally onto the seabed or into the environment that he or she studies. And the same argument applies in support of manned spaceflight, which is also something that keeps being discussed. Some people are asking, what's the point of sending human beings up there into orbit or outer space? And I would even argue that with further improvements in diving technology and becoming more widely available, but also it has become a lot safer over the last few decades, the scope for scientific diving, I would expect, can be expected to increase. I should also say that diving, compared to other techniques that marine biologists have available, is a relatively low cost. Typically, a diving operation is feasible for tens or hundreds of euros, pounds, dollars. A rather expensive expedition was that we had to the Canadian Arctic in 2009, when one diving operation cost about 600 pounds. When we work in Ascension Island, Greece, or Cyprus, it's less than 100 pounds per diving operation, excluding salary. So that is actually quite low cost. If you compare that to the cost of a research vessel plus ROVs, I mean, you're looking at something that is at least an order, at least 10 to often more like 100 times more expensive. And it's impossible to do any ship-based work in that price range. And in comparison, manned spaceflight, I mean, we're looking at least at something above £5 million to put a person into low Earth orbit for doing scientific work. Apparently now with space tourism, that figure has started dropping, but it's for doing serious work. I mean, we're looking still at millions of pounds. Also it should be said that the carbon footprint of scientific diving is much lower compared to doing ship-based work. I mean, any research vessel burns something, even a small vessel will burn something in the range of several tons of fuel per day. I looked at carbon footprint of our Arctic expedition, again based in back in 2009, and the aviation fuel per team member for getting to the Arctic and back from Scotland was around probably 500-600 liters of aviation fuel. And for the whole team and everything, I reckon that we probably burned about 5,000 liters of fuel. This is for an expedition of 3 weeks, I have to say. And this is the daily fuel use of a small research vessel, something like the Sir David Attenborough or the James Clark Ross or the Polar Stern burn something upwards of 50 tons a day. In other words, 10 times that much for a single day. Okay, limitations of diving, I should mention those as well. The main limitation is human physiology and the degree of how many or what can you accept in terms of risks. It is potentially a hazardous activity, and therefore it is regulated by the UK Health and Safety Executive. The most serious issue probably are decompression diseases and accidents. I should also say, touching wood, in now about 27 years of diving, I've never had anything like this in my personal surroundings, and I've never experienced that myself. Also hypothermia, because the seas around here in places like Scotland are cold. There may be dangerous marine life, and often any of these risks can be exacerbated by the remoteness of the study areas. But in many cases, actually getting there, or all the surrounding peripheral work on land or getting to the study site can be as dangerous or more dangerous than the diving itself. Looking again at our 2009 Arctic expedition, the nearest recompression chamber, which is what you want if there is a recompression accident, was in Montreal, Ottawa. And that was about 2 days, at least 2 days of travel away from where we were working, involving several flights, which is the last thing that you want when you have a person having this kind of accident. And therefore, our insurers and the health and safety people at the institution where I was then working imposed a 15-meter depth limit, which is the depth range where such accidents are basically excluded. They're not documented, they just don't occur at less than 15 meters depth. And other limitations can be the level of diving technology that's available, the reliability and performance of equipment, and possibly the cost. But compared again to other techniques, it's rather inexpensive, I should say. Certainly a limitation is depth, duration, that you can stay underwater. Typically not temperature, and I would say that really any aquatic environment that we have on planet Earth is diveable. And now I'm going to talk about a couple of success stories. This man actually is an old friend of mine. He's now in his 90s. George Robert Pettit is one of the forefathers of marine natural products chemistry. He does things that are similar to what our colleagues at the Marine Biodiscovery Center here at the University of Aberdeen, that is Marcel Jaspers, Rainer Ebel, and others are studying. And he pioneered and discovered a number of anti-cancer compounds for marine organisms, and he has been doing this since the 1950s. He started this together with another colleague when nobody else was doing this. And he has published over 800 papers, over 32,000 citations, and a huge H index of 89, probably higher now, dozens of patents. And in the large majority of these studies, and this is why I'm mentioning this, the collections of marine organisms that produce these drug leads go back to diving, providing the starting material. He conducted diving expeditions throughout the world, and he returned to his lab, which was in Tempe, Arizona. And then chemists, a large team of chemists, worked up these materials and found quite a number of interesting drug leads, several of which have made it either to application or to advanced clinical trials. And probably the most famous of these is Bryostatin 1. And it was one of the first major promising anti-cancer compounds developed from any marine organism. And as the name suggests, it's derived from a bryozoan, a marine invertebrate. And it has initial activity that was found against a range of cancers that are really quite deadly to humans. And it has successfully passed phase 2 clinical trials in the US and in Europe. There's a supply problem when you have an organism like this, because in many cases these marine invertebrates are not very large, and if you really have a successful drug, you want a lot of that compound. And in this case, it was addressed by aquaculture. It was actually quite easy to culture this. It's a fouling organism on structures in the sea, and in the future, maybe by genetic engineering, by transferring the pathway, encoding the biosynthesis pathway into organisms that are more easily to culture, in particular microbes or yeast or something like this. And then also synthetic organic chemists were able to synthesize analogs, similar structures that are even more potent than the original lead compound. And now I will return to our Baffin Island expedition in summer 2009, which was the first large diving expedition that I organized. Baffin Island is a very large island. It's about 2.5 times the size of Great Britain. It has only 11,000 inhabitants, and it's not that easy to reach. You may have seen it if you have flown from Europe to the American West Coast to a place like Los Angeles or Vancouver. And our study site was up there in the far north in Pond Inlet, and in fact not in Pond Inlet itself, but at this place Cape Cod, which is about 5 hours by boat. And this is a view of that site. And as you can guess, it is heavily impacted by ongoing climate change. And this was the last preceding expedition led by a Canadian friend and colleague. And this was exactly 6 years later. And there had been actually quite a phase shift in that region. And I think I have to come to an end. The expedition has produced about 10,000 images, over 20 hours of video. We returned with hundreds of herbarium specimens. I'm not going too much into detail. We identified all those. We were able to actually obtain quite a large number of cultures, which is something that we often do in my group, that we apply what is called the germling emergence technique. This is that we collect substratum samples, sediment or sand from the seabed, and then we incubate this in the lab under suitable culture conditions. And usually we see quite a diversity of cryptic macroalgae that you would never see during such an expedition, because your time but also the means in the field are very limited. And then these are further characterized by microscopic and molecular means. And we have published this, excuse me. This paper is available for download. It's open access. These are the main outcomes from this expedition. Now you're probably going to ask, what is the diving like in this place? Well, you use neoprene dry suits, very similar to diving here in Scotland. Not really that difficult. Probably the biggest difference is that you may use full face masks. That's more typical in polar regions. What is mandatory is that you have two divers in the water who are tethered by a rope. This is imposed by diving at work regulations in the UK, and with the long end of the rope going either back to the shore or to the boat. And you have a dive supervisor on the shore and a fully equipped diver on standby on the shore. And then you need a whole shore and boat support team in the case of this Arctic expedition, with 1 or 2 rifles because there's a polar bear hazard in that region. As I said, we had a maximum depth limit of 15 meters because the next hyperbaric chamber was very far away. And however, we managed to spend up to 1 hour in the water. And we managed to conduct a total of 50 diving operations, which is not bad at all. And this is what it looked like. You can see this configuration of the 2 divers, it's Martin Sayer and myself in the foreground, and the long end going to the shore. Finally, this is actually ongoing research in my group. We have a big EU grant, and I should mention my 2 group members, Cleopatra Grammatiki and Eleni Avramidi, who are working on this with me, and we're actually about to go to Cyprus, fingers crossed, maybe at the end of next week, because they're doing their PhDs on this, and because of the pandemic, their work has been badly delayed. And again, diving is key for this work. This is an aerial photograph that I took a couple of years ago in Kuwait. Kuwait is a very dry country, which has no natural freshwater resources, and it makes all of its water needs from the sea. In the old days, they had sailing ships to bring small amounts of fresh water from Iraq, which was about 4 liters per person per day. And now they use about 100 times as much, with a large carbon footprint and also an impact on the marine environment. And this is precisely what we are studying. And this is the place where we want to go at the end of next week. This is the Dekelia desalination plant in Cyprus. And what looks turbid or murky here is actually the brine. If you make drinking water from seawater, you have to put the salt somewhere. In other words, you take out the salt and you dump it back in the sea. This is what virtually all of these plants do. And that has an impact on the marine environment because you have a hypersaline, a very concentrated salt solution. And in many cases, it's also much warmer than the natural seawater, and that impacts marine communities. And now we have funding from the European Commission to, yes? Okay. Yeah. Okay, I will speed up. So this is absolutely key for a number of dry countries. In Europe, this is Cyprus, Malta, Spain, and Greece. And we're looking at what this does to marine life. And among other things, it was found that this impacts seagrass meadows, which are key ecosystems in the Mediterranean, as you will know from our field course, Mediterranean Marine Biology. And this is Eleni diving, and you can actually see that she is kneeling in this brine layer because the visibility is quite poor. And also interestingly, we found that there's a kind of ecological meltdown around these outfalls. There are lots of alien invasive species that have come to from the Red Sea. And maybe I will now very quickly, this is the very last now. We've also been looking at how kelp impacts the speciation of iodine in seawater. And to do so, we collected water samples with large syringes around these large kelp plants, both in California and also in Scotland and in Brittany. And this is myself collecting such samples. And with this, I'm at the end of my talk. Thank you very much. Okay.
So this is the end of the event. If you have any questions, you can ask any of the professors. I just get to them and ask them. And we're going to be in the room next door with the stalls, but otherwise, if you didn't have any other questions, you're free to leave. Thank you for coming, and thank you. Thank you. Thank you.
I don't know if you heard me in the meeting. It's over. Yeah, guys, thank you for being here, and everyone has left actually. They have.